Hi, my name is Patrick, and this is my longer presentation on deep learning BioNLP information from some generated data. In general, the goal of machine reading is that you want to be able to extract mechanisms from the literature, mechanisms in this case being things like the protein family MEK, phosphorylates the protein family ERK, or what are some of, or potentially what are some of the drugs that target BRAF, things like this, which are used in cancer modeling and drug discovery and all sorts of wonderful things. In general, when you can read these things with machines, you can create large dynamical models, which can themselves be, useful, or the models can be, like the network models can be used as priors to various other statistical tools that will help interpret very large data sets that are generated, experimental data sets. It's impossible for a person to read the entire literature, so you can't really have in your head what the entire cellular interactome is like. And for that matter, even having a machine read all of the papers is expensive and difficult to do. So, this NLP is useful. It's also useful for dialogue systems, but it's also not an easy problem. So the academic literature is extremely dense. The grammatical structures can be very complex so that even humans have a hard time understanding them sometimes. There's a lot of possible relations that could come out, and the kinds of things, the ways that you could describe those relationships are also very diverse. So, you have very complex, often very slow models trying to pick up on all this detailed information. But unfortunately, this information is also very spread out. There's, usually a paper will mention a mechanism in detail only, maybe a half a dozen times over the course of many hundreds of sentences. And there's, there's only about like in my experience, like 1 in 10 out of about 1 in 10 papers actually has any useful mechanisms in it at all. So it's very, very sparse. So I decided to try to train a model to predict whether a mechanism exists, right? So that we can save both ourselves as researchers and also our machine readers time. In general, to do this with a deep learning approach, you need to somehow embed the information in the sentence into a way that you can use deep learning tools. Word2Vec is a very valuable tool for this. You can encode the words, but you need to do more than just encode the words. You also need to somehow grasp the grammatical structure. So unfortunately, in this project, I didn't end up actually using that. My code implements the ability to include a grammatical parse. However, it proved to be so slow that I could not actually really run out the code in sufficient time for this project. You also want some kind of named entity recognition, which is essentially picking out things like MEK and ERK and BRAF to say that these are special entities. They have special status, and you should pay special attention to them. And you want to somehow highlight that in your data. So there's existing technology that does does reading. There's like MedScan, which is a proprietary software and probably the most common. Reach and TRIPS, which are things that I've worked with a lot. There's also statistically based readers, which sometimes use deep learning, other times just general machine learning. The results are generally poor due to a lack of labeled data, and arguably due to the challenges of actually encoding the data effectively. It often tends to miss very obvious interactions, or what we as humans would consider obvious interactions, and it doesn't often generalize very well. So existing data sets such as AIMed and BioInfer they're out there, but they're rather small. So as far as I can tell, they have about 1,000 sentences each. They are very high quality. It's a human that actually went in there and picked out the mechanisms that they see. They tend to be very rich, sort of, I'd say, over-representative compared to the literature, right? Because I guess they want to try to portray, well, this is, the most complicated thing you could get. I don't know. What I've produced using Indra, it's a new data set, which you can get using the links that I'll give you. So actually, you can see the links in my project paper. I don't think they were in the slides. But here's are the links that you can use to get the data sets. There's a small and a large. And I would say that in general, it's a large data set, but it's only of medium quality. So it uses multiple different machine, that is to say rule-based readers, to determine what mechanisms are in each sentence. And for that matter, whether a mechanism is in a sentence. So it's better than just, it's not as good as a human curator, but it's better than any one reader. The data set, there's 2 files, only one of which I'll actually be using. So remember that the goal here is to try to predict whether there's a mechanism in a sentence. So here's an example of what the sentence looks like. Right here, you can see it. It has a tag. Entities are tagged in it. So the tau is a type of protein. This, I'm not even going to try to pronounce that, apolipoprotein. It's another kind of protein family, apparently. And it tags those, and it also indicates whether that sentence has a mechanism in it or not. So to do this, I actually use So to parse these sentences, I first had to use Beautiful Soup, basically to strip out these tags the tags here that you see. You can install it just using pip install Beautiful Soup 4. Don't forget the 4. It's really not biology specific. It's just a way to deal with like HTML, XML kind of things, tags within text. I then use the Genia tagger, which is I think which is biology specific. It does various things. It determines the part of the speech, like noun, verb, of each word in the sentence. It also tokenizes the sentence. It also provides chunks, which sort of indicate like generally which part of the sentence each word belongs to, and then also does some fairly basic NER, although I found it to be fairly lacking. You can get this you can do pip install Genia tagger, gets you a Python wrapper, and then you have to install the actual thing itself. You can just click on this link. You can install it from source. If you look at online, you can see the details. Or at my report also goes over it. I did not actually use the blip parser. As I mentioned before, it proved too slow, so I couldn't actually do the data. The Word2Vec is not generally biology specific, but in this case I used a biology specific model, which you can see the link here. It's trained on Wikipedia, PubMed, and PMC, which PubMed and PMC are resources for open resources for biomedical literature. And then, of course, I also used Keras, NumPy, sklearn, Matplotlib, some of the usuals. So first, I extract the text using Beautiful Soup. I basically take the tags out, and I replace them with a temporary identifier right here. So I replace it with this tag, which is like prot1 or prot0. And then I get back the original sentence. I preserve the mappings between the proteins, the temporary tags, and the names. You have to start up the Genia tagger server before you do any of this, but then you can get this client and just parse a sentence. It's quite quick. You get the word base, which is sort of like the basic version of the word, the basal version of the word, the part of speech, a chunk, and the named entity recognition. And then I embed it using Word2Vec. And I also encode the part of speech and the chunk, and I just sort of append these things together. So there's various mapping tools that I use to do this, and you end up with like 200. You do one hot encoding for part of speech and the chunk. The Word2Vec is Word2Vec, and there's 200 slots for those. It's a 200 length vector. The result is something like this. So this is what it looks like. You have each word is a row, and the columns are basically just the data appended. So this is what the embedding eventually looks like. The model I use is a 2D convolutional model. There's one 2D convolutional layer, which essentially just scans down this list 5 at a time, so 5 rows at a time. It looks all the way across. Because each word is sort of its own identity, and so trying to sort of break up the scan lengthwise doesn't really make sense. I then do max pooling and a couple of dense layers, finally producing just a single value, which is more or less just 1 or 0. 0 meaning no relation, 1 meaning yes, a relation. There's a serious problem of data imbalance here. So remember I said that these mechanisms are very sparse. There's like 10 times more negatives than there are positives. So I looked on this resource here to try to figure out how to handle that. One of the things they suggested was defining some kind of class weights in the fit method. So I did that here, sort of more or less based off of what they suggested, but sort of also going off of my own intuition. And then I also defined these two functions based on what they suggested, which are a better metric of success rather than just accuracy, right? So if I just guessed all zeros in this case, I would get a 90% accuracy, even though I'm really just completely wrong. I then trained the model. As you can see, it didn't do stellar. The validation, so the training fit very nicely, but the validation did not. Strangely enough, the small data set does seem to have trained at first, but the large data set did not. That was actually the reverse from what I expected. But in both cases, we seem to overfit very quickly. I think this has to do with just the diversity of kinds of input, but also perhaps with this data imbalance, which you can see here, right? We do very well on the specificity, which is more or less a measure of the quality of the negative results, but we do extremely badly on the sensitivity, which is more or less a measure of how well we guess positive results. So yeah, and I created a confusion matrix. Again, you basically see the same story here, right? We do well at predicting negatives, but very poorly at predicting positives. So the lesson learned is that embedding text is hard, especially this very complex text. Loading the Word2Vec file, interestingly, is actually very slow. It's a very big file, takes up a lot of RAM. It's also hard to parallelize because you can't just copy that file into multiple threads. You'd have to create a service or something. You need a lot of data to produce a useful classifier. I think maybe if you had more data, you could possibly do better. But the data imbalance also creates all sorts of problems. It's also a bit difficult to debug exactly why the data imbalance, what its effects may be, or even if that is the effect. Perhaps a simpler approach to embedding would have actually been better in this case, given the simple outcome that I'm looking for. In fact, you could probably make pretty good predictions just by counting the number of entities. In fact, here the data set already was filtered down to things that had at least 2 entities, and that actually filtered out, I think, 10 down to 1% of what all was out there. So that already gets you a long ways. And then, how to embed these sentences in a useful manner for deep learning is actually still very much an open topic of debate. So thank you very much. The YouTube URLs probably don't help you much because this is one of my YouTube URLs. Thank you.